Hello guys, I am Sachin. I am going to take about case study on Azotobacter species. Azotobacter species is a nitrogen fixing bacteria which is isolated from the rhizosphere plants. Rhizosphere plant means soil surrounding the plant roots. It was first discovered by Dutch microbiologist and botanist Martinus Bergenic, who was one of the founder of environmental science. He was discovered in 1901. Now we are going to see about general characteristics. Azotobacter species is a gram-negative bacteria. It is oval or spherical in shape, size 2 to 10 mm long and 1 to 2 mm wide. It is aerobic respiration, optimum temperature 20 to 30 degrees Celsius, optimum pH about 7.0 to 7.5, endospore is absent, capsule is present, commonly found in soil, water, sediments and plant roots. It is motile. These are the common Azotobacter species. I will read few on Azotobacter baigenic, Azotobacter paspali, Azotobacter vinelandi, Azotobacter agilis. Now, Azotobacter species recommended crops like commonly known crops are recommended like rice, wheat, millet, cotton, vegetables like potato, onion, tomato, brinjal and other vegetables, sunflower, mustard and flower crops. It shows 10 to 15% yield increases. Azotobacter takes place in nitrogen fixation. Nitrogen fixation in Azotobacter species because it enhances nitrogen availability in soil by converting atmospheric nitrogen into ammonia, which can be readily taken up by plants. This reduces the need for synthetic nitrogen fertilizer, which means chemical fertilizers. Biofertilizer. Azotobacter also takes place in biofertilizer. In biofertilizer, Azotobacter improves soil fertility and enhances plant growth. It provides a balanced and organic source of nutrients to plants. Soil improvement. It improves the soil structure, aeration, water holding capacity. They also enhance nutrient availability in the soil in the root zone, promoting better plant growth. Crop inoculation. Azotobacter applies in the soil inoculant to specific crops or seed, especially legum. It boosts nitrogen fixation and overall crop yields. Environmental cleanup. Now we are going to see about environmental cleanup. Azotobacter cleans heavy metal and other pollutant which present in soil. They help detoxifying soil and make it more sustainable for agriculture use. Biological control. It also produces antimicrobial substance which inhibits the growth of certain plant pathogens. It is used in integrated pest management strategies to reduce the need for chemical pesticides. Organic farming. Farmers who interested in organic farming, they use Azotobacter because it is chemical free farming and environmental friendly. Research and biotechnology. Azotobacter used research related to nitrogen fixation, which has broader implication for understanding this crucial biological process and enhancing nitrogen fixing capability in other plants. greenhouse and nursery agriculture. It controls environments such as greenhouses and nurseries where soil health and nutrient management are essential for plant growth. Reforestation and land reclamation. Azotobacter can play a role in reforestation projects and land reclamation effort by aiding in the establishment of vegetation in nutrient poor soils. Okay, now I am going to explain nitrogen cycle. First, nitrogens are present in the atmosphere. It is a bivalent triple bond. It was break down by the lightning with the help of lightning. But lightning was not occur anytime. So the alternative method is called nitrogen fixation. Nitrogen fixation. First, fixation means converting the element into its usable form. Microorganisms like bacteria and blue-green algae present in soil can help to fix nitrogen. It is also called as nitrogen fixing organisms. In microorganisms, compounds like nitrite, ammonium are present. It breaks down the nutrition and the plant takes it to synthesize protein. Now I will explain about nitrogen fixation. Plant requires protein, but nitrogen has ammonium acid, it has protein. So the nitrogen was break down by lightning. It is not occur anytime. So the alternative method is nitrogen fixation, converting an element into its usable form. Microbes present in soil break down the nitrogen and it was taken by plants as a protein to synthesize protein. Now we are going to see nitrogen cycle. Nitrogen cycle means cyclic flow of atmospheric nitrogen to the living world and back into the atmosphere, which means it has two methods. Atmospheric nitrogen means first method rhizobium bacteria for legumes. Second method nitrogen fixing bacteria and blue-green algae. Nitrogen was break down by bacteria in soil, then it takes by plants. The plant was eaten by animal and the animal gets nitrogen. It was trapped in their body. After the animal death, they are decomposed in the soil and the nitrogen was released. It has another method called denitrifying bacteria, which is pseudomonas. Denitrifying is occurred by this bacteria. This bacteria decompose and release the nitrogen into the atmosphere. Denitrification and decomposition of both take place in the atmospheric nitrogen. They help the nitrogen back into the atmosphere. Now we are going to see isolation of Azotobacter species. Martinus Beijenic was the first to isolate and describe Azotobacter. Azotobacter cells. It is not present in the rhizoplane. Rhizoplane means root of the plants. Rhizosphere region is surrounding the plant, which means rhizosphere is present in the surrounding the plant region. It is isolated from the rhizosphere soil by serial dilution method. It has three methods: serial dilution method, spread plate method, or pour plate method. Culture medium used for isolation is Asbys mannitol agar. Microscopic characteristics of Azotobacter. Gram staining. It is pink color, oval or rod to spherical shape, chain, single chains or clumps. Motility test, also called as hanging drop method. Actively motile rods due to peritericoous flagella. See, this is the Azotobacter species. It looks in pink color, rod shape. Now we are going to see cultural characteristics of Azotobacter species. In Asbais manita lagar, the growth of Azotobacter species is large, good and luxuriant. Azotobacter species like Carcocum shows brown to black colonies, and Azotobacter species Beijenic shows yellow colonies. Chemical characteristics. Catalysis catalase test is positive, oxidase test is positive, urease test is positive, indole test is positive, methyl red test is negative, Vogus phosphorus variable, citrate utilization test is positive, gelatin hydrolysis negative, starch hydrolysis positive, casein hydrolysis positive. Overall Azotobacter species biochemical test is gram staining positive, motility test is motile, catalase test is positive, nitrate reduction positive, pigment production positive, H2S production positive, urease test positive, citrate test positive, utilization of carbon source positive, glucose positive, fructose positive, mannitol is negative, indole test is positive. Reference. I hope you all understand about my topic. So thank you.